and that of the covering and lining epithelial tissue. We highlighted that the glandular epithelial tissue, as the name implies, is associated with forming the structure of our glands. We have two types of glands. We say glands will produce and release a secretory product. We have the what? Exocrine glands, the duct-containing glands, and then we have the ductless glands, are what? Endocrine glands that produce and release hormones, bind to surface receptors or bind to receptors. They can also be intracellular and regulate cellular activity. We're focusing primarily on the covering epithelial tissue. As the name implies, it covers and lines surfaces of the body. It helps establish boundaries between different compartments within our body. Some of the examples of the covering and line epithelial tissue, here we have the epidermis, the outer layer of the skin. We'll see that the epidermis is made up of a type of tissue that we call our what? Keratinized, stratified, squamous epithelial tissue. So it's a type of stratified. We said we talked about the covering and line epithelial tissue. We use a naming system. The first name will denote the number of what? Cell layers. It can either be simple or stratified. Stratified meaning multiple cell layers. Simple meaning single layer, implying that all those cells are in direct contact with the what? Basement membrane. And the second name represents the what? Morphology, in which we have our squamous, boido, and the what? Columnar stage cells. So the example, our covering and lining epidermis. Also said our membrane, which are associated with the ventral body cavity, line the walls of the body cavity, and the outer surface of the organs within that respective body cavity. They're made up of epithelial tissue. And then also we said we find epithelial tissue line the lumen or the cavity of our organs, the hollow organs, as well as that of our blood vessels. We identify the epithelial tissue line the lumen of our blood vessels being that of our simple squamous epithelial tissue, forming what's called the what? Endothelium. Forming what's called the endothelium, which plays a major role in blood flow with regard to changing the caliber or the diameter of the lumen of the blood vessels to increase resistance or to decrease resistance. Now we list some general functions. Of the epithelial tissue, we said some of the general functions will be that of what? Protection. Protection will be provided by that of our what? Simple? These what? Stratified epithelial tissue. Other functions will include that of absorption, filtration. What do you mean? Filtration is a type of what? Active transport? Passive transport, and we're doing what? Moving what? From high concentration to low? Yeah. And filtration, we're driving down a what? Pressure gradient. We're using a hydrostatic pressure to press water and solids from the what? Capillaries into the what? Tissue space. That interstitial fluid. Did we mention that yesterday? Yeah. Okay, so the capillary walls are porous. Adjacent endothelial cells have small gaps between them, and that within that capillary, hydrostatic pressure will force the solids and the water from the circulation into the tissue space, giving rise to the interstitial fluid. So we have protection, absorption, filtration, secretion, excretion, and then also when you think about your skin, we have what? Sensory receptors. We have proscenium corpuscles, minor corpuscles, nerve endings that allow us to respond to stimulation to the surface of the body. General functions. Also in your reading, they highlight some characteristics that highlight the structure or the structural anatomy of our epithelial tissue. So it has some characteristics that highlight the structural anatomy of the epithelial tissue, which helps distinguish it from some of that of our other primary tissue types. So make sure you take a look at some of the characteristics that highlight epithelial tissue. One such characteristic of our epithelial tissue is as a tissue type, epithelial tissue exhibits what's called cellularity. It exhibits cellularity. And when we say cellularity, epithelial tissue consists of tightly packed cells. Tightly packed cells that will form sheets of tissue, which will do what? Function to cover and line the what? Surfaces of the body. So as a tissue, structurally cellularity, it consists of tightly packed cells. And it has very little extracellular material. This is in contrast to connective tissues. In contrast to tissue, connective tissue has very little cells and a very extensive extracellular matrix. So one characteristic of our epithelium, it consists of tightly packed cells, and it has very little extracellular material, and these tightly packed cells help our epithelial tissue to form sheets of tissue, sheets for tissue, which allow its function to cover and line the surfaces of our body. Are we clear on this? I'm trying to say here? We're saying as a tissue type, it consists mainly or predominantly of cells, very little extracellular material, which is in contrast to our connective tissues such as bone tissue, cartilage tissue, tendons, ligaments, which have a very extensive extracellular matrix and very few cells. Epithelial tissue consists of tightly packed cells. In order to get these tightly packed cells to form the sheets of tissue, another physical property or structural property, I should say, for our epithelial tissue is that it consists of special junctions. So it has specialized and we talk about specialized junctions. Epithelial tissue is associated with cell junctions that link adjacent cells together. So we link adjacent cells together to what? Form our sheets of what? Tissue. So they're associated with cell junctions or membrane junctions, and these cell junctions will function to link adjacent cells together to form these sheets of tissue that can line the surfaces of our body. So we have densely populated cells. We don't want them to be spread apart. We want them to be what? Together or exhibit cohesion. So these cell junctions or membrane junctions will link adjacent cells together. As you think about these cell junctions associated with the epithelial tissue, in chapter 3, they highlight them, but also they mention them in chapter 4. And keeping in mind, we're doing 2 chapters in tandem, 3 in lecture, 4 in lab. But we have, for example, a type of cell junction, CJ, for cell junctions associated with epithelial tissue. We have tight junctions. We have what are called desmosomes. And then we have gap junctions. So these are the type of cell junctions which will help to link adjacent cells together, form these sheets of tissue, epithelial tissue, that will cover the surfaces of the body. Are we okay with that? The cell junctions that link adjacent cells together are formed by what? What forms these cell junctions between adjacent cells? What links them together? Molecularly, what does it?
Membrane protein.
Membrane protein, exactly. So the walls are membrane proteins. As we say, the proteins are the big daddy of the cell, so to speak. And by this I'm saying the proteins do most of the what? of a cell, and we use the term proteome to see what? Structure and what? Function of the cell, because it determines the types of what? Proteins that a cell will produce. Are we okay with that? And based on the type of cell, proteins the cell makes, we determine the structure and function of the cell. If you take a look at just an example of a couple of these cell junctions, we have a tight junction. A tight junction will form biomembrane proteins to represent two adjacent cells. And the tight junction will involve a class of membrane proteins called glutins and claudins. These are transmembrane proteins that span intercellular space, fused together to form a junction between adjacent cells. So we have involved our glutins and claudins. These are just the names of transmembrane proteins associated with two adjacent cells. They will span the intercellular space. You see the intercellular space, talking about the space between adjacent cells. They will span the intercellular space. So they span the intercellular space as they together and they form an impermeable junction or an impermeable seal. As these transmembranes from two adjacent cells fuse together in direct contact, they form an impermeable seal or junction that will prevent the passage of material from one compartment to a what? Another compartment. Are we okay with that? So it's establishing what? Boundaries between different environments in the body. So let's say, for example, these are two epithelial cells lining the lumen of the intestine, lining the lumen of the stomach. Underneath, we have our vasculature or blood vessels being able to absorb materials from the intestinal tract into the circulation, deliver it to the cells and tissues of the body. If they're lining the lumen of the stomach, we know that the stomach is associated with what? Digestive enzymes, harsh digestive enzymes that can damage tissues. Are we okay with that? As we chemically break down food, also within the lumen of the intestine, in addition to digestive enzymes, E for enzymes, we also have microorganisms associated with the digestive tract, and to prevent the access of these damaging digestive enzymes from gaining access to other compartments of the body, to prevent these microorganisms from gaining access to the what? Circulation, we form these tight junctions to compartmentalize one environment from the body to another. Are you okay with that? Is this making sense? If these microorganisms were able to gain access to the circulation, this would cause what? Wide systemic what? Infection or sepsis, which will lead to shock and death if not treated almost immediately. So by having these tight junctions, this helps to compartmentalize different environments within the body in order to get material from one compartment to another compartment. In order to get material from one compartment to another, what will we require? What would the cells require? If we have these tight junctions preventing the passage of material from the lumen, the intestine, into the circulation. So if we want to get something from the circulation, something from the lumen of the intestines into the circulation, what do we need? The question get across. Say again. In order to get material from one compartment to another compartment, we we'll have to have a specific what? Transport protein, because since substances cannot gain free access between the interface, if we want to transport something from one compartment to another, we need a specific transport protein to carry out that process. Are we okay with that? So we have our type junctions as a type of cell junction to link adjacent cells together. In addition to linking cells together, it is serving a function to form an impermeable seal to protect the body from different types of environments within our body. Another example of a cell junction would be a desmosome. All three of these are highlighted structurally and functionally in Chapter 3. So make sure you take a look at Chapter 3 with regard to these cells. A desmosome is an anchoring junction and it is to link adjacent cells together and also to distribute stress or mechanical stress applied to the epithelium or to applied to the tissue. So distribute stress applied to our tissue as you take a look at the structure of a desmosome. These are two adjacent cells, and on the lateral border of each cell will have a disc-shaped complex of proteins called a plaque. So we have a plaque on the lateral border of two adjacent cells. It's just a disc-shaped complex of proteins. From the plaque, we have what are called cell adhesion molecules, or proteins called our cadherins. So we have these cadherins these are transmembrane proteins that extend from two adjacent cells, and they will interdigitate to what? Link adjacent cells together. So we have radiating from the plaque, we have our cell adhesion molecules, our CAMs. So the acronym CAM is for cell adhesion molecule. An example would be the cadherins. And these cadherins will interdigitate linking adjacent cells together. So this is forming our desmosome. To reinforce the desmosome to the opposite side of the cell, radiating from the plaque, other side of the cell, we have our intermediate filaments, and these intermediate filaments will reinforce the desmosomes, also distribute the stress or mechanical stress applied to the skin, to the epithelium. You think about your skin, we have mechanical stress throughout the course of the day. You bump up against the surface, that's applying stress to the tissue. To prevent that tissue from tearing from just the minor mechanical stress, we want to reinforce the tissue, or the cells that make up the tissue, I should say, by these desmosomes. So these are intermediate filaments. Intermediate filaments are one of the three type of what? Cytoskeleton elements. We have the microtubules, microfilaments, and the intermediate filaments, and they help to reinforce the desmosome anchoring junction. Make sure you take a look at the gap junction in Chapter 3 and make sure you describe the structure and the function of a gap junction. So we have cellularity, cell junctions. Another characteristic associated with epithelial tissue that is said to exhibit what? Polarity Our book says it exhibits polarity. And when we say, polarity, what do we mean with regards to epithelial tissue? Oh, wow. Remember, it's a generic term. It just means opposite. So we say polarity. Nathan. Yeah, okay. When they said polarity, what do they mean? I'm trying to get your name right now. Right here. Your name again? Donna. What do we mean when we say polarity? Right, two sides. And what are the two sides called? We mentioned it before. We said a what? Right.
Apical and basal.
So we have two surfaces associated with our epithelial tissue. We have the apical surface and the basal surface. Why when we say the apical surface, what do we mean when we say the apical?
It's the
So it's the upper surface. Is that what you said? It's the upper surface. It's the exposed surface. When we say it's exposed surfaces of the body. So it's the upper surface. It's exposed to the surfaces of the body, and it's unattached. It's the unattached surface of the epithelial tissue. So it's the upper surface. It's exposed to the surfaces of the body. So this would be the apical surface of the epidermis, exposed to the surface of the body. The apical surface would be that exposed to the lumen of an organ. Are you okay with that? That's the upper surface, the exposed surface. That's what we mean, exposed to the surfaces of the body, and it's unattached. The basal surface will be the what? Lower surface of the epithelium. Not upper, it's lower, so it's the lower surface. It's unexposed, and it's the attached. The polarity is just a generic term, means opposite. You have the upper surface, the lower surface, the exposed, the unexposed surface, the unattached, attached. Are we okay with that? So that's all we mean when we say polarity. Also, when we take a look at the polarity or the differences between two surfaces, look at them structurally. The apical surface, this would be the apical surface. So this is apical, exposed to the surface of the body. This is the basal surface. We can see structural modifications on the apical surface that we will not see on the what? Basal surface. We mentioned two types of modifications that we see associated with the upper surface, and those two types of modifications will be that of what? Jermaine? Microvilli. Why do we have microvilli? You're Donna too, right? So I got two Donnas. Okay, Donna. You're Donna, right? All right. What is microvilli? What does microvilli do? What's the function of microvilli? Exactly. So it increases the surface area of a cell, and we see involved in absorption. We want to have the invagination of the plasma membrane to increase the surface area, so we can increase the rate of what? Diffusion. Are you okay with that? Structurally, we said the microvilli are made of the what? Microtubules? Micro what? Filaments made up of the protein what? Actin. We can have on the apical surface, we can have microvilli. In the book, they talk about what's called a brush border. Have you guys seen that in the book? They say a brush border, which is just a dense population of the microvilli. So a brush border is a term you want to be familiar with. Or we can have the cilia. We said cilia are longer hair-like appendages that are modal, or have the capacity of what? Movement. And what's responsible for the movement of the cilia? We said it the other day. Tatiana. Trenton, go ahead. The 9+2 arrangement of what? Tubules, but the microtubules interact with what? Brick. Oh, no, you're Courtney Banks. Right? Sure. We said that the cilia have a capacity of movement. What's responsible for the rhythmic movement of cilia, which allows cilia to move substances along the surface of the tissue? What's responsible? Great, go ahead. Binding and what? Kinesin, we said that the microtubules interact with those what? MAPs, the microtubule associated proteins, dynein and kinesin. They have an ATP site, which is a catalytic site, which allows them to bind to hydrolyzed ATP. Use that energy and go to conformational change. They pull and release on the microtubules, causing the cilia to move back and forth, which allows them to move substances along the surfaces of the tissue. So we have these modifications on the apical surface. On the basal surface, we said they're attached to a support matrix, and the support matrix that underlies our epithelial tissue called the what? Basement membrane. So they're attached to the basement membrane. They're attached to the basement membrane, the support matrix of our epithelial tissue. Our basement membrane is a support matrix. Well, we'll say it's a thin sheet of non-cellular material. So it's non-cellular. It consists primarily of fibers or fibrous proteins. So it consists mainly of fibers. The basement membrane will function to what? Support and anchor the epithelial tissue. So it underlies the epithelial tissue. ET for epithelial tissue. It helps to support, anchor epithelial tissue. It also helps, as it supports epithelial tissue, it helps it resist tearing from what? Mechanical stress. So it helps it resist tearing. The basement membrane will also provide or help epithelial tissue establish boundaries. And as we take a look at the basal surface, it's attached to the basement membrane by way of what's called a hemidesmosome. We have a hemidesmosome. This will be a plaque. This is half of a desmosome. We have collagen proteins that will interact or anchor the basal surface of the cell to the basement membrane. So we have a hemidesmosome, which will anchor the basal surface, BS for basal surface, to the BM, don't have any space, basal surface to the what? Basement membrane. Okay with that? Basal surface to the basement membrane. Structurally, the basement membrane says to have two layers. So we have it going horizontal and vertical. The two layers of the basement membrane will be that of what? Jessica. We have what's called the basal lamina. So the superior aspect of the basement membrane is called the basal lamina, which is made up primarily of glycoproteins. The glycoproteins are secreted by the epithelial cells, and the basal lamina of the basement membrane is considered to be the epithelial component of the basement membrane. So the membrane has two lamina or two layers. The superior layer is called the basal lamina. It consists primarily of glycoproteins that are produced and released by the epithelial cells. So the basal lamina is referred to as the epithelial component of the basement membrane. What we're trying to say here. So it's the epithelial component of the basement membrane. The basal lamina will also function as a filter to regulate what enters the epithelial tissue regulate what enters into the epithelial tissue. Then we have the lower layer of the basement membrane. It's called the reticular lamina. The reticular lamina consists primarily of collagen fibers and the collagen fibers are secreted by fibroblasts, cells associated with what? Connective tissue. So the collagen fibers are secreted by fibroblasts, which are associated with connective tissue. So the lower level or lower lamina of the basement membrane is considered to be connective tissue component. So the basement membrane, basal lamina, glycoproteins produced and released by the epithelial cells, filtered to regulate what enters the epithelial tissue. It's the epithelial component of the basement membrane. The lower lamina is called the reticular lamina. It consists primarily of collagen fibers, which are, well, actually they release the collagen, and the collagen will self-polymerize. We don't want to think of the cell release the big old fiber. And it's the tissue component of the basement membrane. So we listed three characteristics: cellularity, specialized junctions, polarity. Another characteristic is that epithelial tissue is supported by connective tissue, and it's supported by connective tissue via the what? Matt, what's your question? A part of the extracellular matrix part, because the basement membrane is extracellular. That's extracellular material. You say extracellular, you just say it's not cell. The collagen fibers are part of the membrane of the reticular lamina. Okay, you got that? Another characteristic associated with epithelial tissue is that it's supported by connective tissue, and it's supported by connective tissue by way of the what? It's supported by connective tissue by way of the what? Say it again. The reticular lamina. So it's supported by connective tissue by way of the reticular lamina of our what? Basement membrane. Mr. Chambers, you okay with that? So it's supported by connective tissue. When we said whenever you find what? Epithelial tissue, you're going to find connective tissue because it's a component of the what? Basement membrane. All right. Also, epithelial tissue is avascular. Say it's avascular. Vivian, what do we mean when we say avascular?
Avascular? Oh, it doesn't have blood vessels.
Exactly. It's avascular, so blood vessels. So Carlos, how does it get its nutrients that it needs? If epithelial tissue is avascular, how does it get the nutrition that it needs for the cells to? That's deep. David, the question is, if epithelial tissue is avascular, it lacks blood vessels, how does the tissue get the nutrients and all the material it needs to carry out its function? Danny? Daniel. Sorry, Daniel. So we're asking, if epithelial tissue blood vessels is avascular, how does the cells of the tissue get the nutrients that it needs to carry out its function? Matt Allen. It's not Miss Ramirez, is it? Oh, Miss Ramirez. Renee. Miss Simmons. Why? You got your notes right in front of you. I'm not going to ask you, bro. Diffusion from what? Blood vessels where? All right, you got your little. What are these you guys have? What is that? Man, you guys got easy. Yeah. You're absolutely correct. Via diffusion, because in the underlying basement membrane, which is connective tissue is highly vascularized. So via diffusion, blood vessels supplying the reticular lamina of the basement membrane, we have material diffusing from the underlying capillaries of the connective tissue to the epithelial tissue. Are you okay with that? So it's avascular, this nutrient via diffusion